Welcome to our webinar. Today is the World Cancer Day. February 4th has been announced as the World Cancer Day. In every country of the world, people remember or they are getting reminded that cancer is an unconquered disease. Almost every one of us either know the person, family member, a friend, or a colleague affected with these health problems. And this is the most feared diseases, not only because of its uncertain outcome, but also because of treatments that are being applied in cancer patients. Cancer is a disease that doesn't discriminate. It affects famous and unknown, young and old, people living in poor countries, and also people living in wealthy societies. There are many organizations that their work has been dedicated to education, but most of them work to increase funding for cancer-related research. And you know them by name, whether you live in Great Britain, Poland, Austria, Germany, or in America. You know these organizations. I know in the United States there are famous foundations or organizations that organize sporting events or cultural events to raise research for cancer. So let's look on who are the sponsors of World Cancer Day. The first names that pops out, you can see it on this screen. The major partner of this program is Bristol Myers Squibb pharmaceutical company, also Amgen and Roche. So you can see that these companies are promoting themselves and also looking for more funds in doing research. But do they really need our funds? Let's look at Bristol Myers Squibb and their financial status. As you can see, that this company spends almost twice as much on marketing than on research. Do they really need our funds, which we already dedicate to cancer research through our taxes and, of course, participating in different fundraising activities? Do pharmaceutical companies really need more public funds? Here, look at this chart, which presents the average profit margins of 5 main industrial sectors. And these are the data for 2013. You can see the pharmaceutical business has the highest profit of banking business, car maker business, or oil and gas and media industries. The highest profits can reach about almost 50%, 42% profit. The lowest one, 10%. On average, they are making the most money on producing medicine that, as we know, in case of cancer, does not cure this disease. Cancer is the disease, as mentioned earlier, that affects everyone, affects known people, famous people, rich people, poor people. And recently, the Vice President Biden in the Vice President of in the United States was affected by death of his son, and the death was caused by brain cancer. And following this tragic event, the President Obama announced during his 2016 State of the Union address that United States government will start new anti-cancer initiative and brought this initiative brings some connection to the initiative of putting a man on the moon that was announced by President Kennedy many decades ago. So this initiative under the name Cancer Moonshot will be led by Vice President Joe Biden. It is a very remarkable event and very much needed event. But let's look whether the direction of this initiative really give the hope that we will find the cure for cancer. So I look at the main task and main focus of this initiative, and then you can see that one of the focus will be a initiative sharing national clinical data regarding this disease, and also the federal government will put money on the project in gene sequencing. And it is interesting that this is not genetics, which is the responsible for majority of cases of cancer. Cancer in about 80% is caused by environmental effects, but our diet, toxins in our environment, and other environmental factors. So what's really new in this cancer Moonshot initiative? So we can see that, from the beginning, this direction does not point in the right way, because we need the government support not for exercising already tried or already executed avenues which led to nowhere, but we need the government support for trying new untested ideas and very bold ideas that were not tried before, and they have to be promoted and they have to be supported. Because the old path, trying the same approaches with chemotherapy, radiation, developing drugs that target individual cancers, not common cancer mechanisms, have been tried for almost 6 decades. The war on cancer continues since 1970, but approaches to cancer were tried many decades earlier. So, but now, based on our research, based on the study which we've been conducting for the last 2 decades, we see that there is a chance to control cancer, and there is no time to waste. We need to this new direction that gives us hope in developing effective way of controlling and beating cancer. So how cellular medicine, what are the main aspects of cellular medicine that allow us to have this hope and believe that we are on the right way? First of all, we need to start from understanding the common nature of cancer. What are the common mechanisms that are involved in cancer growth and spread when it comes to cancer of the uterus and skin cancer and colon cancer and many others? We need to understand which are the common mechanisms so we can develop measures against them, and then we can succeed in many types of cancer. The other aspect is selecting the biological targets that are critical for effective control of cancer. And we all know that the most deadly stage in cancer is metastasis. This is the process when cancer invades many organs in the body, and at this point there is no cure, there is no effective way to stop it. Unfortunately, the interest in studying metastasis is very low, and the funding for metastasis research is very limited. But this is a critical target necessary to controlling cancer. Cellular medicine also looks at the alternative ways of developing therapeutic approaches to commonly practice pharmaceutical chemical molecules, because cellular medicine focuses on using micronutrients, natural substances that are combined in synergy, natural substances that do not have side effects. They are both safe and effective in many stages of and many different health problems. So how do we approach to in our studies that can lead to effective control of cancer? First of all, selecting right scientific path for controlling cancer. And we know that pharmaceutical approaches, conventional approaches, look at individual single biological target in cancer. And this is why we have a treatment for a specific type of breast cancer, for prostate cancer, different treatment for skin cancer. And there is a direction that focuses on individual specific types of cancer and specific treatments. And also what I mentioned earlier, ignoring this direction ignores metastasis, the process that kills cancer. What are the measures developed by conventional approaches? These are, as you know, drugs, antibodies, and chemotherapy drugs, and also radiations procedure. How a cellular medicine approach differs. We are looking at the common aspects in cancer development, and by identifying those common aspects, we can develop approaches that target multiple biological steps at once. So in this aspect, by understanding the mechanism that underline tumor growth, underline formation of new blood vessels that feed tumor, which is angiogenesis process, and mechanisms involved in metastasis, we can focus, we can attack all these key mechanisms at once, and we can do it using the synergy of several natural substances. This is something that we called multi-nutrient synergy approach. So let's look in more detail how our comprehensive approach to cancer looks like. So in looking for the common mechanisms of cancer and those targets that we can approach with natural substances, we are looking at the natural means of preventing destruction of connective tissue, this extracellular surrounding that accompanies such processes like cancer growth, cancer metastasis, and angiogenesis. Because in all these three aspects, the cancer cells need to destroy their surrounding in order to create place, more room to facilitate the regrowth. They need also more room to pave the way for growing blood vessels, new blood vessels, and also they need to pave the way to move from one organ to another. So from one side, we are looking at the natural substances that can inhibit this destructive process, inhibit enzymes that destroy, digest connective tissue. On the other hand, we are looking at the ways how we can increase the integrity and strength of this connective tissue surrounding cancer cells, so we form a so-called barrier around tumors, around cancer, and that way we can prevent expansion of cancer cells in the tissue. So we look at the substances that can support this process. We also look at the natural substances that can kill cancer cells in a selective way without killing healthy cells. This is what we know. It's different than cancer chemotherapy, where the substances, chemical substances, very powerful toxins are applied, which kill both healthy and cancer cells. Natural substances work in a different way. They can be selective. They can eliminate cancer cells, but do not destroy or harm healthy cells in the body. And the third aspect, many natural substances that can support our body in these functions that I mentioned earlier, they also have positive effect on enhancing our immune system function, and therefore they support our body in fighting cancer and in attacking cancer cells. So what are the natural compounds that support our body in all these three aspects? So let's look at the natural substances that can curb enzymatic destruction of connective tissue. These are enzymes that belong to the class of proteolytic enzymes. Enzymes digest protein. In this case, they digest components, natural components of connective tissue, such as collagens and elastin and other compounds. These enzymes are known as urokinase and also known as MMP2 and MMP9, which activity has been associated with aggressiveness of cancer. We also know natural inhibitors of those enzymes. And here you have a list of the most active one, which is amino acids lysine, vitamin C, EGCG, which is an active component of green tea, also curcumin, quercetin, resveratrol, and also the components coming from cruciferous vegetables are effective inhibitors of destruction of connective tissue, which are the nutrients that can support strength, synthesis, and integrity of connective tissue. These are, first of all, vitamin C. Vitamin C, which can regulate how much collagen, this main connective tissue component, is produced in the body, and also it assures that the collagen produced has optimum structure, which is essential for maintaining optimum function of this protein. Vitamin C is supported by amino acids lysine, proline, which are the components of collagen, but also vitamin B6, flavonoids, and copper and iron. Now, what are the nutrients which can support our body in killing cancer cells without harming healthy cells in the body? These are nutrients that can exercise so-called pro-apoptotic effects, supporting natural death of cancer cells. And there is also quite a long list of those, and which include vitamin C, again, this epigallocatechin gallate, the components of green tea, N-acetylcysteine, and plant components, curcumin, quercetin, resveratrol, and many others. Here, I would like to show you one of our most recent results that confirms the impressive efficacy of micronutrients against cancer. Some of you who follow our research know that we have been developed nutrients composition that can effectively control about 50 types of different cancer, 50 types of different cancer cells. And this approach is effective in inhibiting growth of tumors and also curbing metastasis. Here I want to show you the recent experiment where we tested the efficacy of this nutrient composition in ovarian tumors. And on the upper picture, you can see the examples of tumors ovarian tumors that developed in animals that were kept on controlled diet. These were large tumors, and we also observed that these animals had developed lung metastasis. In contrast, you can see on the lower picture that animals that had the same diet but supplemented with a specific micronutrient mixture these animals did not develop ovarian tumors. In the first set of experiments, we had 6 animals per group, and we didn't see any tumor developed in the animals taking this micronutrient supplementation. So we said we need to try it again. Maybe it was, mistake or something wrong went with the experiment. So we had another set of 6 animals that were put on this micronutrients supplemented diet. And among 6, only 1 animal developed small ovarian tumors. So you can see that only 1 out of 12 animals developed tumors when these animals were consuming diet supplemented with this selected micronutrients. This is a remarkable result because ovarian tumor is very aggressive and it's very difficult to treat. So during the course of our research, we came to conclusion that there can be 2 sets of micronutrients that show documented and show efficacy against cancer, and we call it as a synergy 1, which contains multiple nutrients that contains vitamins, amino acids, trace elements, and plant extracts that have been shown effective against a variety of cancer types. And another synergy compound that contains only selected plant extracts such as curcumin, green tea extract, quercetin, and others. So there are 2 sets of micronutrients that show this very potent anti-cancer efficacy. The other aspect that we recently started to investigate that relate to cancer relates to the interaction between cancer cells and healthy cells in the body. We all know that cancer cells do not exist in our body by themselves, but they are surrounded by normal cells, and therefore by the nature of them, healthy and normal cells interact with each other. So we were interested to see whether proximity of healthy cells to cancer cells have positive or negative effect In other words, whether healthy cells can promote growth of cancer, or maybe they have inhibitory effects on cancer. So we started to study the interaction between these two types of cells, and also to study how micronutrients can interfere with this biological communication that exists between cells and decrease any pro-cancer effects that may occur between these two cells. So on this slide, I would like to share with you some of the results. Maybe slide look a little complicated, but I will try to guide you and see and show that actually the results are not complicated at all. So we tested the expression of key enzymes that destroyed connective tissue in cancer cells, and also in cancer cells combined with normal cells. So everything what you see in blue color, the blue color blocks represent cancer cells only, the level of enzymes expressed in cancer cells only. The red colored columns show the change in the activity of these enzymes when cancer cells were combined with normal cells. So you can, by just looking at the page, you can see that in general, the activity of all this connective tissue digestive enzyme increased, so that normal cells actually can accelerate the destruction of connective tissue in the vicinity of cancer cells. We looked at the enzymes that we know are associated with cancer growth, aggressiveness of cancer, such as MMP2 and MMP9. And you can see the activity of those enzymes increased by the presence of normal cells. When it comes to the enzyme with the symbol plow, this is urokinase, the activity was almost the same, but the another compound which is called PLA-T, which is the inhibitor of plasmin, this urokinase, decreased. So then all this explains that interaction of normal cells and cancer cells results in enhanced anti-cancer activity. So these were the results that we conducted in vitro with isolated cells, but we want to learn more about it, and especially look at how micronutrients affect this, I would say, cancer-promoting destruction of connective tissue in a living organism. So we did the experiment in which we injected the combination of cancer and normal cells into a mouse. We used breast cancer cells. And then the animals, after injecting cancer cells plus normal cells, were put on a different diet. One, it was a standard diet. The other diet contained this nutrient mixture that I mentioned earlier, which contains different natural compounds, and then diet with nutrient mixture number 2, which contains mostly plant extracts. And we look in particular how this exposure to different diets affects growth of tumors in these mice, and also whether it affects the expression activity of these key enzymes that are involved in destruction of connective tissue in malignancy. So you can see that exposure of or putting mice on this different diet resulted in inhibiting tumor growth, and quite significant, about around 50%. There was no much difference between the efficacy of these two nutrients, diets. Now, then we were looking at how the activity and how the expression of the specific enzymes involved in cancer process changes depending on the diet. And here I can show you the results, the efficacy of this nutrient mixture number 2, which contains which is the combination of plant extracts. So you can see that after adding normal cells to breast cancer cells, the activity of MMP2 and MMP9 increases, which confirms the earlier results obtained from in vitro experiment. MMP increased by 30%, MMP9 a lot, by 80%. Urokinase decreased 10%, which is basically, we can say that the expression didn't change. And this inhibitor of urokinase increased 50%, which means that the enzyme was active, or activity increases. And this is what happened when we compare the activity of those, or expression of this enzyme after adding normal cells, where this normal cancer cell interaction took place. And now look what happens when in mice that were consuming diet containing this nutrient mixture number 2. The MMP2 activity decreased 50%. MMP9 the expression increased 80%. So it's complete reversal. Urokinase activity 95% decreased, and expression of the tissue inhibitor normalized. So from this experiment, you can see how powerful are these nutrients, and that these nutrients can exercise their effects not only directly on cancer cells, but also on this interaction between normal and cancer cells that results in tumor promoting activities. It is a very important finding, which shows that micronutrients are very powerful in affecting not only processes directly associated with cancer cells metabolism, but also this interaction between normal and cancer cells. Many of you contact us or ask question how we can enhance efficacy of micronutrients. Most of the studies that we conducted so far related to cancer preventive potential of micronutrients, that they are that the intake and their presence in our body is very important in preventing, slowing down cancer development. However, we also looked at more at the therapeutic aspects of micronutrients. And a few years ago, we conducted this study testing the efficacy of micronutrients working in synergy versus individual micronutrients when they are applied to already developed tumors. In this case, we studied the breast cancer tumors, and we allowed those tumors to develop to the certain stage that we could identify them and we could measure their size. And then we monitored growth of these tumors in 3 groups of animals. One group was placed on control diet. The other group had the same diet by enriched with green tea extract, a very known anti-cancer compound. And the third group had the same diet by supplemented with nutrient synergy. You can see that on a control diet, the growth of tumor continues steadily. Green tea extract had some inhibitory effect on growth of tumors, but nutrient synergy, the combination of micronutrients, was most effective and resulted in slowing down of growth, progression of tumors almost 90%. So we know that these micronutrients are effective when they are applied also at the late stages of cancer development. Some patients have been using natural approaches, not always with expected results. So therefore, we need to analyze why the animal study results cannot be extrapolated to human situation, and there are several multiple aspects that relate to it. But in this particular case, we need to take into account that people always, most often, apply non-conventional other approaches, including cellular medicine approach, at very late stages of cancer development. Stage 4 of cancer, which also involves metastasis, and in cases where all other modalities, conventional modalities, were already applied, and the body is devastated by several rounds of chemotherapy, radiation, and many other treatments. This has an impact on the function of intestinal function, and it results with compromised absorption of micronutrients. So we can take them, but they are not absorbed and utilized in the body. Also, those treatments in advanced stage of cancer results in the compromised immune system. And another aspect is that at the stage of despair, people start applying everything what comes across. And sometimes it involves approaches that are not effective, and sometimes then even cause harm. So this is why the results are not always or not as expected. Many people also ask us about intravenous vitamin C. So I would like to address this issue shortly. So vitamin C has very well documented anti-cancer activities. How does it work? From one side, vitamin C can strengthen this natural barrier around cancer cells, around tumors, that prevent cancer cells from spreading in the body. So it forms the barrier. On the other hand, it has been shown that it's very effective in killing cancer cells. So it has a direct effect on cancer, and it is safe to healthy cells in the body. There are many formulations of vitamin C suitable for intravenous delivery available with different doses, and people are using that. The efficacy of intravenous vitamin C increases when it is applied together with oral intake, and there have been clinical studies showing that for maximum efficacy, intravenous application of vitamin C have to be accompanied by oral intake. So we also studied the effects of vitamin C itself on cancer, and here I can show you the results of our study that confirm its efficacy in forming the barrier around tumors, and therefore preventing spread of cancer cells, preventing its invasion in the tissue, but also relocation to other organs. In this study, we used a special type of mice. These are mice that do not produce vitamin C, like humans. And we divided these mice into two groups, One group was kept on a normal diet. The other group had a diet supplemented with vitamin C. And we observed that after injecting breast cancer cells, the growth of breast cancer tumors was very aggressive in mice that did not have vitamin C in their diet. When we look at the cross-section of those tumors, we were not surprised. You can see it on the left-hand side that these tumors had any limitation in their spread in the tissue. You can see there is no barriers, cells are separating, so they could freely spread in the body. And this is in contrast to breast cancer tumors that developed in mice supplemented with vitamin C. You can see this white, pink, blue color barrier surrounding the tumor mass, which shows and it confirms this effects, very important effect of vitamin C in controlling cancer. And in addition to its effect on limiting the growth of tumors, we observed that vitamin C supplementation inhibited metastasis in mice about 70%. So it confirms this anti-cancer efficacy. Now, what about the efficacy of vitamin C in killing cancer cells? In order to explain it, I have to talk about how vitamin C is transported inside the cell. So there are several pathways that vitamin C gets inside the cell. One of them, which occurs when vitamin C is, here is labeled IAA, ascorbic acid. Vitamin C is at low concentration outside of cells. In this case, it just simply diffuses through the cell membrane. The other way of transporting is through specific channels. And in this case, ascorbic acid can enter the inside of vitamin C using sodium channel. This is exchanging with the sodium. Vitamin C goes inside and sodium goes outside. So it's an exchange with sodium. The other way of vitamin C to get inside the cell is using the same channel that glucose uses, sugar. So in this case, the vitamin C is transported on this sugar used gate as dehydroascorbic acid, oxidized vitamin C. So in this case, it competes with glucose for the same path. And when it enters the cells, then both dehydroascorbic acid, this oxidized form of vitamin C, and other forms are converted to vitamin C. So this is what happens in general. Now, there is a difference how vitamin C behaves inside the cancer cell and inside the healthy cell. And this is what I'm going to explain on this slide. So let's start with the healthy cell. And then we have both ascorbic acid and dehydroascorbic acid entering the cell using different gates, different transporters. But when this form get inside the cancer cell, they all are converted to ascorbic acid. And the healthy metabolism of the cell continues. Now, what happens when vitamin C gets inside cancer cell? So vitamin C gets inside the cancer cells as dehydroascorbic acid, this oxidized form of ascorbate. And in cancer cells, due to the specific cancer cell metabolism, dehydroascorbate cannot be converted to reduced form of ascorbic acid, to real vitamin C. And therefore, cancer cells accumulate lots of this oxidized vitamin C, which has a detrimental effect on cancer cell metabolism and lead to cancer cell death. So the difference in cancer cell metabolism and healthy cell metabolism is responsible for different effects of vitamin C on the cells. How much vitamin C is needed to kill cancer cells? So there have been many studies trying to explain this and investigate this problem. What is known is the most human cancer cell types are killed when the concentration of vitamin C is less than 10 millimolar. Some types of cancer cells need much higher concentration, even above 20 millimolars. Okay, how does it translate to vitamin C intake? So what is known, the infusion of vitamin C in amount of 100 gram, which is by some standards a lot, can result in this 20 to 25 millimolar plasma levels, which is detrimental to cancer cells, which kills vitamin C, which kills cancer cells. For some people, 100 gram is it's an enormously high level, and people are concerned with safety of such high doses. When it comes to safety of vitamin C, this is one of the most safe substances. However, it can be used by people who have a genetic problem. They suffer from glucose 6 dehydrogenase deficiency, and high doses of vitamin C may cause hemolysis in blood. People with kidney problems and people who have iron overload. Otherwise, safety of these vitamins have been documented in clinical trials, in many case reports, and even doses 100 or 150 grams a day infused few times a week show to be safe. And its IV delivery was tested up to 10 months of, frequent weekly use. And this was shown to be safe, generally safe. And these studies were not conducted in some backstage operations, but they were done at McGill University in Canada, Thomas Jefferson University, University of Iowa, and famous for vitamin C study and application, Jordan Clinic. Many studies, published studies, also document improved quality of life when vitamin C is applied intravenously in cancer patients. It has been shown that its use is safe in conjunction with other supplements, and this even includes some chemotherapy agents with some, this is not recommended, and it hasn't been tested to be used with radiation. So we don't know what is the efficacy of intravenous vitamin C when applied together with radiation. So these are very powerful effects, anti-cancer effects of vitamin C when it is used as a single agent. And in this case, it has to be used in very high amounts, very high quantities. So we were interested in comparing the efficacy of vitamin C in high doses as a single agent to vitamin C used in much, much lower doses, but combined with other micronutrients working together in synergy. So here I show you this comparison. And in this table, you can see the efficacy of vitamin C provided by intravenous injection, efficacy on tumor growth, on occurrence of metastasis, and on lifespan. This was an animal study. So this study was conducted at the National Institute of Health in the US, and the application of vitamin C was 4 grams a day, 4,000 milligrams a day. And in this study, tumor growth, there were different tumor types tested, was inhibited between 30-56%. There is no clear report how metastasis was affected, and the lifespan was extended by 20%. How these results compare when vitamin C is used in synergy with other micronutrients. In this case, in a synergy, vitamin dose was 20 to 30 milligrams a day, much, much lower. However, the impact, the efficacy was much higher compared to single vitamin C application. Tumor growth could be inhibited by up to 92%. It's tremendous. Then metastasis, up to 100%. It can stop vitamin C in synergy with other, can stop metastasis, and we observed that lifespan of these animals can be expanded by about 30%. So these are remarkable results. And I would like to, at the end of my talk, I would like to quote one of the recent correspondence that we received, acknowledging our scientific approach to cancer. And it has been written by a doctor. And here you see the quote, "Thank you again for your work. I was introduced to your work through the documentary, The Truth About Cancer. Unfortunately, in the documentary, legitimate researchers like yourself are mixed in with pseudo-doctorates that never graduated from any postgraduate school." And then she says, "I see patients and know that the truth of the matter is skewed on both sides of the argument. I wish it were so easy as a yes or no question when it comes to conventional medicine usage. It is not." And this was written to us, in January, end of January, before the World Cancer Day. But it shows that health professionals, other scientists, recognize that our approach against cancer gives us a chance to control this disease 100%. We need to continue our study. We know which mechanisms need to be affected for the effective control, and we believe that we will be 100% successful in achieving victory over cancer. So yes, we can win cancer, and we can win it with nutrient synergy. Thank you for your attention.